Good evening, folks. Hope everybody is doing well tonight. This is going to be part 3 in my lecture series on the digestive system in humans. We may have a part 4, we may not. Depends on how long it takes me to get through the remaining content. I am assuming at this point that if you're watching right now, you've probably already seen parts 1 and 2 already posted to this channel. If you have not seen them yet, please go back and watch them before you watch this part so that you have the necessary background for where we're going from here. If you are in my anatomy and physiology course, please make sure that you've gotten the handout from the Blackboard site so that you have important vocabulary and a summary of the notes. And I also strongly recommend you keep either your lab manual or your textbook handy to follow along with some diagrams along the way so that you have some visuals to go with the things that I am describing to you here. Now, up to this point, we have been taking a tour of the digestive tract going from mouth down on from there. We have been through the mouth, the oral cavity, the pharynx and its various components, the stomach, and the small intestine. We have also begun discussing the accessory structures attached to all of this. We talked about the teeth, the tongue, the lips, and the salivary glands as components that are going to assist with the action of the digestive system. So for this lesson, I want to start focusing on the lower digestive tract. But before we can leave the small intestine behind, in order to understand its function, we're going to have to talk about a few accessory organs that contribute to it. Now, the small intestine, as you hopefully remember from last lesson, is where chemical digestion is completed. That within the duodenum, we largely finish the chemical breakdown of most of the parts of your food that were not previously broken down in the mouth and the stomach. But to facilitate that, we're going to require a lot of digestive enzymes and secretions. Most of those do not come from the small intestine itself. Rather, they are secreted by other organs and then delivered to the small intestine. Now, I should point out this is an exocrine rather than endocrine function. Remember, endocrine function is the release of hormones generally into the bloodstream, whereas exocrine function is the release of secretions either onto the free surface of the body or to the inside of the lumen of a digestive cavity. In this case, the cavity being the lumen inside of the duodenum. But our secretions that are going to be going in here are going to come primarily from two organs that you probably already know of, and one of which you've talked about in a previous lesson, namely the liver and the pancreas. And when we're talking about the liver, along the way, we're going to end up saying a few words about one of its accessory structures, the gallbladder. So that's where we're going to begin here. Let's start by talking about the liver and the gallbladder. It makes sense to discuss them together because they are an integrated structure that are, well, they're integrated structures that are doing basically the same task to facilitate chemical digestion. Now, the liver is an organ found in the upper right quadrant of your abdomen. It's resting up against the inferior surface of the diaphragm, so it's the very top of the abdominal cavity. The liver is your largest visceral organ. It's the largest internal organ in the body. Comes in at right around 1.4 kilograms, 1.3 to 1.4 in the average adult human. Liver is a very busy organ. We're going to talk about its functions shortly, having functions that go well beyond digestion, although the digestive functions are the ones we're most concerned with right now. Now, in terms of its structure, again, I would strongly recommend you look at a diagram here. It's rather difficult for me to model in description here, and you all know what my art looks like by now. But we divide the liver up into lobes, L-O-B-E-S, just like the brain is divided into lobes. The liver has 4 distinct lobes. We have a left lobe and a right lobe, which are joined together by a band of tissue called the falciform ligaments, F-A-L-C-I-F-O-R-M, falciform. Sort of tucked behind and beneath them, we will find the other 2 lobes, the quadrate lobe, Q-U-A-D-R-A-T-E, and the caudate lobe, C-A-U-D-A-T-E. Now, the quadrate and caudate lobes are separated from the left lobe by a fold of the greater omentum. The quadrate lobe is going to be separated from the right lobe by the gallbladder. The gallbladder is tucked between the right and quadrate lobes. And the inferior vena cava forms the boundary between the right lobe and the caudate lobe. Runs upward, passes between the two lobes on its way. There is an opening on the inferior surface of the liver, right in the vicinity of the left lobe, called the porta hepatis. Porta, P-O-R-T-A, hepatis, H-E-P-A-T-I-S. The porta hepatis is the opening that admits a number of things that come into the liver: its blood supply, its nerve supply, and ducts to carry away its secretions are all going to pass through the porta hepatis. Now, if I look inside of the lobes of the liver, we mentioned 4 lobes. We have left and right lobes, quadrate lobe, and caudate lobe. If I look inside of them, I will find that internally, my liver is going to be divided up into 8 segments. Now, you might be naturally thinking, okay, 4 lobes, 8 segments, 2 segments per lobe. Nope, not quite that simple. The segments of the liver are an internal division that is completely separate from the external divisions. So it's not at all unusual for segments to overlap more than one lobe of the liver. So don't think of the 8 segments as corresponding neatly to the 4 lobes. The division's a little bit messier than that. Most of the lobe level division of the liver is basically sections of the liver that are separated from one another by the attachment or passage of other structures. And so internally, the liver has a completely separate division system. Each of the segments of the segments of the liver, I don't know what my problem is here. Okay. Each of the segments of the liver is actually functionally independent of the other segments and can be individually removed without harm to the rest of the liver. And in fact, the liver is a highly regenerative organ. It recovers quite easily from damage. If an individual liver segment is injured and has to be removed, or is destroyed and has to be removed, it can actually regenerate from the rest of the liver within 6 to 12 months, as long as the rest of the liver remains healthy. You can function with about half of a functional liver and still carry out all the essential functions. The entirety of our liver is surrounded by a capsule of dense, irregular connective tissue. It provides external protection for it, with the exception of a small area on its superior surface. The small area that is not covered by the capsule is called the bare area, B-A-R-E, and that is pressed up against the diaphragm. So the diaphragm is providing it with its protection. In terms of its cellular composition, cells of the liver are made up, are called hepatocytes. H-E-P-A-T-O-C-Y-T-E. Prefix hepa, H-E-P-A, means having to do with the liver. So hepatic portal system, hepatocytes, hepatic artery, and so forth. That porta hepatis that I mentioned, that opening in the inferior surface of the liver, is going to admit the external blood supply coming in via the hepatic artery and the hepatic portal vein. That's hepatic portal vein is hepatic, H-E-P-A-T-I-C, portal, P-O-R-T-A-L, vein. The hepatic vein, which is not the same thing as the hepatic portal vein, also goes through the porta hepatis. Hepatic artery brings in blood supply to the liver. Hepatic vein takes away blood supply from the liver. It's called drainage from the liver. Hepatic portal vein has another function. We'll get to that momentarily. And we're also going to have the hepatic nerves entering along this pathway through the porta hepatis. And finally, we're going to have the hepatic ducts, D-U-C-T-S, exiting the porta hepatis, carrying the secretions of the liver, specifically bile, toward the duodenum of the small intestine. Now, your liver is a busy, busy organ. It's one of the busiest organs in the body, in fact, after the brain. The liver has more than 300 documented individual functions. But you can group them into a couple of broad categories, things that the liver are doing. First and foremost, and perhaps most importantly for our digestive purpose, the liver is engaging in bile production, B-I-L-E. Every day, a healthy liver secretes approximately 600 to 1,000 milliliters of bile. Bile is a yellowish green fluid made up of bilirubin, bilirubin, cholesterols, fats, and several hormones, most notably a substance called lecithin, Lecithin. The bilirubin components of the bile, from which the word bile is derived, you might recall that substance. Bilirubin is a yellowish green pigment that is released when red blood cells are broken down. The liver recycles the bilirubin from the red blood cells and converts it into bile. If the liver is not functioning properly, bilirubin can accumulate in your tissue, and that can lead to a characteristic yellowing of the skin and the whites of the eyes that we call jaundice, Jaundice. Jaundice is often an indicator of a misfunctioning liver or malfunctioning liver. It can be an early sign of liver failure from cirrhosis. It can be an indicator of liver cancer. It can be an indicator of hepatitis, 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 which is a virally induced inflammation of the liver. All those things can potentially leads to failure of the liver to produce bile properly, and as a result, bilirubin accumulates in your tissue. Incidentally, whenever you get a blood transfusion of the wrong blood type, you all know that can be lethal within a couple of days' time. If I give you type A blood and your blood type is B, and the reason for that is because when you get the wrong kind of blood, you have a type 2 hypersensitivity response. Remember from our immune discussion, a transfusion reaction. Your body rapidly hemolyzes, that is, breaks down the bad red blood cells that have the wrong markers on them, and that releases huge amounts of bilirubin into your bloodstream. The consequence of that is the liver gets overwhelmed. It is unable to process all the bilirubin. The bilirubin in turn goes into your kidneys, clogs up your kidney ducts, and you end up going into acute kidney failure. The main reason people die from transfusion reactions is acute kidney failure as a consequence of built up bilirubin that the liver could not process because there was just too much of it at once. All right, then. So you got bile produced from bilirubin, lecithin, cholesterol, fats, and a couple of regulatory hormones, as well as a number of minerals that we collectively call the bile salts. Bile is responsible for neutralizing and diluting the chyme that has entered the small intestine. Bile is a basic compound. Remember, the chyme that is going into the duodenum is acidic. So the bile serves to dilute it, so it's not as concentrated, and it also serves to neutralize its acidity. But the most important function of bile in terms of digestion, oh, by the way, we have to do that neutralization, because that helps with pancreatic function. The secretions of the pancreas do not work as well in a highly acidic environment, and therefore the bile is helping to make that environment more neutral for them. But the other really big thing that bile is doing is the emulsification of fats. Emulsification, F-I-C-A-T-I-O-N. Emulsification is whenever you break anything up into smaller bits and disperse it throughout a solution. Fats, you probably recall, are insoluble in water. That makes digesting them a little bit tricky, because they tend to form clumps. They tend to clump together. They tend to not have a lot of surface area relative to their volume. That's good old hydrophobic exclusion. You might remember that from Bio 1, that your nonpolar molecules, like fats, try to interact as little as possible with a watery polar environment. So they form clumps or beads or shapes like that that minimize their surface area relative to their volume. Problem with that is that makes it harder for digestive enzymes to access those fats. And this makes fat digestion more complex than that of proteins and carbs, which are more water soluble. So what emulsifiers do is they break apart materials and disperse them throughout the liquid of the solution. Basically, it takes what bile does is takes big clumps of fat, emulsifies them, divides them into little tiny beads of lipids, and spreads them out throughout the liquids in the small intestine. That has the effect of breaking it into more easily digestible chunks and gives you more surface area overall for the digestive enzymes that process lipids to work with. So, secretion of bile is a major function of the liver. Without it, fat digestion, and indeed all digestion, would become a little trickier. Another function, storage. The liver is a major storage organ for a bunch of different nutrients. Your hepatocytes will regularly extract small amounts of sugar from the bloodstream to prevent hyperglycemia, that is excessively high blood sugar. When your blood sugar goes up, your liver begins slurping up sugars from the blood, taking glucose out, and then it takes that glucose and stores it in the form of glycogen. You probably remember that word, Glycogen. Glycogen, remember, is the storage form of glucose in animal bodies. It is animal starch, if you will. And our livers store a certain amount of glycogen, basically as a way of lifting excess glucose from circulation. Your liver can keep a glycogen reserve of up to somewhere about 800 and 1200 kilocalories, and can maintain it for between 18 and 24 hours. So it can gradually secrete it and release it into the bloodstream as blood sugar levels go down. Whenever you do your carb loading before your big workout, you got the big game tomorrow, you're going to be running a lot. So what do you do the night before? You have that big carb-rich meal, lots of potatoes and pasta and stuff. What you're doing is artificially inflating your blood sugar so that your liver says, uh-oh, we better store up some of this, converts a lot of it into glycogen, and then gives you a glycogen reserve that you can draw upon when you're engaged in your big game or helping your friends move up a flight of 17 stairs or whatever. 17 stories. Let's go 17 stories to make it as bad for your friend as possible. But so our liver stores glycogen. That is a major thing that it does. It also stores fat. A certain amount of fat storage in the liver is a good thing. You're probably all aware that fatty liver, the condition of excess fat storage in the liver, often as a consequence of cirrhosis and a consequence of diabetes. These are both problematic, but a certain amount of fat storage in the liver is normal. The liver also stores certain minerals, most notably iron and copper, which give the liver its distinctive reddish color, the iron in particular. Also, the liver stores a large number of vitamins, in particular, vitamins A, B12, D, E, and K are all stored by the liver. Here's a fun fact for you. Liver, you probably know, some of you have probably eaten liver at some point in your life. It's a good old British classic meal, liver and onions. And some of you might know that organ meats like liver are very healthy for you because they're very rich in vitamins and minerals. Lots of protein and lots of vitamins and minerals in there. So eating liver is a great source of vitamins, in particular vitamin A. Now, that said, should you happen to find yourself lost in the Arctic Circle one day and you're hungry and you're looking for a meal and you see a polar bear over there, naturally, you're going to break out your spear and kill the polar bear for dinner, right? That's what you would do. Don't eat the liver. You might be thinking to yourself, ooh, lots of good nutrients in that polar bear liver. Do not eat the liver. You're going to find that a lot of marine animals in the Arctic, effect of that metabolism is that they store very high amounts of vitamin A in their livers, in fact, a toxic amount of vitamin A. If you eat a polar bear liver, you are likely to die from vitamin A toxicity, which is hard to get in most other situations. All right, so nutrient storage of several different vitamins, of a couple of minerals, and of fat and glycogen. All right, that's an important liver function. While we're talking about nutrients, let's talk about nutrient interconversion. 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 What I mean by that is that the liver is very good at converting one nutrient into another. That is, it can change certain things into other things that your body requires. Among the things that the liver can do, it can take excess amino acids and deaminate them. Deamination, D-E-A-M-I-N-A-T-I-O-N, deamination. Be careful with that word. It is not deanimation. Deanimation is when you kill Donald Duck. Deamination is when you break down proteins. Think amino. Deamination. So normally your body prioritizes proteins primarily for use in building other proteins. Namely, we digest proteins and our cells say, ooh, those are amino acids we can use to build other proteins with. But if you've got excess proteins, your liver will deaminate them, break them down, in order to make ATP. We'll put them through a process of deamination for ATP synthesis. You can do that a little more indirectly than glucose. You can also convert deaminated proteins into glucose through a process called gluconeogenesis. 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 Gluco for glucose, neo means new, genesis to create. Creates new sugars, right? So we can deaminate proteins either to make things for ATP synthesis or for gluconeogenesis to make glucose. You can also convert excess proteins or carbs into fat. That's the thing the liver can do. Excess proteins or carbs can be converted into fat for storage. Fatty acids can be broken down by the liver to make ATP through a process called beta oxidation. Beta, we use the Greek letter beta, Greek equivalent of the letter B. Find it in the insert symbol part of your word processor to break down the fatty acids and make ATP that way. You can also break down fats and use their fatty acids to make phospholipids. You can convert fatty acids from triglycerides into phospholipids, which your cells use to make their membranes. And finally, your liver can also take lactic acid that was a byproduct of muscle metabolism. Remember that your muscles, when they are not making enough energy via aerobic respiration, are forced to switch to anaerobic respiration and begin converting glucose into fermentable byproducts in that way. And lactic acid is the waste product of that. Lactic acid can then be transported to the liver, where it can be converted back into glucose through gluconeogenesis. So all of these are nutrient interconversion functions of the liver, things that the liver will basically, require, one nutrient to be transformed into another. Another big function of the liver, detoxification. You already knew this. This is probably the best known function of the liver. Your liver is your primary organ of detoxification. Hepatocytes contain many enzymes that are used to break down harmful compounds that have entered your body. A lot of times what this means is taking a harmful compound and breaking it down into more harmless compounds that are easier for the kidneys to remove. For example, a lot of your cells make ammonia as a waste product of metabolism. Ammonia is a difficult substance for the kidneys to excrete in humans. Therefore, the liver converts it into urea. Enzymes in the liver will detoxify ammonia, breaking it down into urea, which is much more easily excreted by the kidneys. You will also find that excess drug compounds will be detoxified and broken down. Keep in mind your liver does not know whether a drug you take is good for you or bad for you. All your liver knows is that thing right there is foreign. That's a molecule my body doesn't make. So when your liver detects drugs in your system, whether it's illicit or perfectly legal drugs, your liver is going to break them down eventually and say, all right, let's break that down and detoxify it. Because as far as your liver is concerned, any foreign molecule is a toxin, whether it's actually hurting you or not. This is why drugs wear out after a while. This is why you take a dose of a drug and 6 hours later or whatever, you need to take more of it because your liver has broken down that drug. Over time, as you probably know, your liver can build up a tolerance for specific toxins. It can say, okay, this particular toxin right here is coming into my body regularly. I need more capacity to process it. And the liver, what it will do is the hepatocytes will add more smooth endoplasmic reticulum and more enzymes to break down that particular toxin. After a while, the liver gets better at processing a given toxin if it's regularly exposed to it. This is why after a while, you build up a tolerance to alcohol. If you drink regularly, you need more to get the same buzz, right? Or if you are on pain meds long term, you might build up a tolerance for them, and after a while, you need a higher dose of that pain med in order to get the same effects, which is why people on long-term pain management often switch up their pain drugs from time to time to prevent tolerance buildup. So you can find that you will eventually get better at processing particular toxins. One of my favorite stories in this regard involves people who handle poisonous snakes for a living. And you might think, why the heck would that be my job? It's because we need their venom to make anti-venom. If we want to make an anti-venom against snake poison, what we do is we inject a wee tiny amount of snake poison into a large-bodied animal like a horse. The horse makes antibodies against it. Then we extract blood serum, that is plasma from the horse, used to call it horse serum, to make anti-venom. And that that serum will then contain antibodies against that venom, which we can use to treat people who have been bitten by a poisonous snake. But we need the venom to do that. So snake handlers naturally are risking being bitten by poisonous snakes all the time when they're trying to capture the snake and hold it and make it bite down on a flask so that we pump the venom out, milking the snake, they call that. Well, people who handle snakes regularly will often deliberately expose themselves to tiny, tiny bits of snake venom, little tiny diluted injections of snake venom, specifically so their livers become accustomed to it, become used to breaking it down, and therefore gives them a tolerance in case they get bitten for real. Similar thing happens quite by accident to beekeepers. People who keep bees and naturally are going to get stung periodically as part of their task, eventually their livers get better at processing the venom in bee stings, and they build up a tolerance to that. Now, some things the liver cannot detoxify. There are some molecules that are too big or too complex for the liver to break down. Those tend to be the poisons that are most dangerous to us. Some things the liver can't detoxify, but what it can do is secrete them into bile. So some molecules that the liver cannot fully break down will instead be secreted into bile and released from the liver that way, dumping them into the intestinal tract where eventually they'll be released along with your wastes. Okay, other functions of the liver. Phagocytosis. You remember that word, phagocytosis, is when a cell gobbles up and engulfs another cell or a chunk of food or a bacterium or cellular debris or whatever. The liver contains a lot of phagocytic cells, macrophages and so forth. Among other things, the liver phagocytizes worn out blood cells, in particular red blood cells, which it then uses the bilirubin from them to make bile. But it also breaks down worn out white blood cells, cellular debris that has entered the liver from other parts of the body through the bloodstream, from other dead cells, and bacteria. The liver actually has an immunological function in that it phagocytizes bacteria that come into it through the blood. And finally, the liver has lots and lots of synthesis functions. The liver synthesizes a very wide range of proteins. Among others, it synthesizes most of your blood proteins, the albumin that makes up the viscosity of your blood plasma, the various globulins that circulate in blood plasma, the clotting factors, the fibrinogen, heparin, the anticoagulant. All of these things are examples of proteins synthesized by the liver. It's not an all-inclusive list, but it's an example. So, a lot of different functions. You probably recognize that most of those things I said were actually umbrellas for wide ranges of functions. Nutrient interconversion is a bunch of different things. Detoxification is a bunch of different things. Synthesis is a bunch of different things. Storage is a bunch of different things. Plus, of course, we have phagocytosis and we have production of bile. These are all things the liver does. As you can see, it's an extremely busy organ. You need your liver for survival. You do not last long without one. Fortunately, as I said before, you need about 1/2 of a functioning liver to carry out all these functions. Between 1/3 and 1/2 of a liver will do most of what your body needs. So you've got a lot of spare capacity, just like you have a lot of spare kidney capacity. Now, you might have noted, a lot of those functions of the liver that I just enumerated are functions that involve nutrients, right? The liver converts one nutrient into another. It stores excess glucose or vitamins or minerals. It is building things, synthesizing things, which therefore means it needs amino acids. Bit of a tongue twister there. So, given all of that, your liver wants access to nutrients. In fact, your liver gets first crack at all the nutrients your body takes in. Whenever your small intestines have finished digesting stuff, the first thing that's going to happen once those nutrients get absorbed from the small intestines is they are heading straight to the liver. The liver gets first crack at all the nutrients that come into your body so it can set aside whatever it needs for its synthesis and storage functions. Now we achieve that with a special network of blood vessels called the hepatic portal system. Remember, a portal system is any place in circulation where you have two consecutive capillary beds, that you absorb things in one capillary bed, you route them to another capillary bed, and only then does the blood move on. There are not a lot of places in the body where you have portal systems. Specifically, you've got the hepatic portal system, and you have the hypothalamo-hypophyseal portal system between the hypophysis or pituitary gland and the hypothalamus of the brain. But the hepatic portal system is the largest portal system in your body, and its job is to give the liver first access to nutrients from digestion. Here's how it works. We start off in the small intestines. We have finished the breakdown of our food, we have completed chemical digestion, and now we got a whole lot of nutrients circulating in the chyme. Simple sugars, amino acids, fatty acids, and so forth. Those things diffuse across the walls of the small intestine. They're collected by the villi and microvilli in the small intestine and enter the capillary beds associated with the villi. That's the first set of capillary beds in the portal system. So through those capillary beds in the small intestine, we are going to collect all of those nutrients. Then that nutrient-rich but oxygen-poor blood is going to leave the small intestines via the hepatic portal vein. The hepatic portal vein will then carry it into the liver through the porta hepatis. From there, that blood is routed to structures in the liver called portal triads, T-R-I-A-D, triads. Now the word triad means a group of three. And it's kind of a misnomer because when portal triads were first discovered, we thought they had three parts, hence the term portal triad. We have since learned that a portal triad actually, in fact, has 5 parts. The 5 things that go into the portal triad. We have one branch of the hepatic portal vein. Once it gets to the liver, it splits into lots and lots of branches, going to lots of portal triads. So you've got one branch of the hepatic portal vein bringing in nutrient-rich blood. We have a branch of the hepatic artery, which brings blood to the triad. We have lymphatic vessels, which are there to drain excess fluid and also to collect lipids. You might recall one of the functions of the lymphatic system is the absorption of lipids. We have the common, we have a bile duct, sorry, not the common bile duct yet. We have a bile duct, which is there to collect bile. And we have a branch of the vagus nerve, good old cranial nerve 10, which regulates the activities of the triad. So the blood from the hepatic portal vein comes into the triads. The blood from the hepatic artery also comes into the triads. Hepatic artery is delivering oxygen-rich blood from the heart. Hepatic portal vein is delivering oxygen-poor but nutrient-rich blood from the small intestines. That blood gets mixed in the portal triad, and then it empties into hepatic sinusoids. Hopefully you remember sinusoids, S-I-N-U-S-O-I-D. Sinusoids, you might recall, are the capillaries with the largest openings in their walls. So these are your liver capillaries. Through those hepatic sinusoids, we're going to exchange oxygen and nutrients with the tissue of the liver. This is the second capillary bed in our portal system. And in that way, the liver grabs what nutrients it needs and sends the rest on. Finally, the deoxygenated blood, carrying any nutrients the liver didn't want, exits the liver via the hepatic vein, not to be confused with the hepatic portal vein. So our pathway in the hepatic portal system is going to be small intestines, well, capillaries of small intestines, I should say, hepatic portal vein, portal triad, hepatic sinusoids, and then hepatic vein. All right, sound good? And in this way, your liver gets access to all the nutrients it requires before any other part in the body gets access to them. Now you might have recalled that one of the big things the liver is doing is it's synthesizing and transporting bile. Bile, as you saw before, is essential for digestion. You need it to emulsify fats and to neutralize other digestive juices present in the duodenum. So we need to get the bile from the liver to the duodenum of the small intestines. Now, bile is synthesized by hepatocytes. The various cells in the liver synthesize bile using bilirubin extracted from red blood cells. That bile is then carried away from the hepatocytes through tiny channels called bile canaliculi. C-A-N-A-L-I-C-U-L-I. The word canaliculi, singular canaliculus, means little canal, little channel. So these tiny little channels, the bile canaliculi, carry blood away from the hepatocytes that make it to the portal triads. You might recall that the portal triads also contain a bile duct, also called a hepatic duct. The hepatic or bile ducts collect the bile. They converge from little ones onto bigger and bigger ones. Ultimately, they all converge on two large ducts called the right hepatic duct and the left hepatic duct. The left and right hepatic ducts then direct bile into another single large vessel called the common bile duct. Sorry, the common hepatic duct, excuse me. The common hepatic duct is then going to join another vessel, the so-called cystic duct, C-Y-S-T-I-C, to form the common bile duct. So our pathway thus far is hepatocyte, bile canaliculi, portal triad, hepatic ducts or bile ducts, converging on right and left hepatic ducts, then common hepatic ducts, and then the common hepatic duct and the cystic duct are going to join together to form the common bile duct. Now, the common bile duct is then going to exit the liver through the porta hepatis. It's going to travel to the small intestines. At the small intestine, the common bile duct is going to merge with the pancreatic duct, P-A-N-C-R-E-A-T-I-C, which, as you can probably guess, the pancreatic duct is carrying secretions from the pancreas. Those two ducts, the pancreatic duct and the common bile duct, are going to fuse together at the hepatopancreatic ampulla, or ampulla. That's a bit of a mouthful. Hepatopancreatic, one word, H-E-P-A-T-O-P-A-N-C-R-E-A-T-I-C, hepatopancreatic, ampulla, A-M-P-U-L-L-A. So those two ducts fuse together at the hepatopancreatic ampulla. The hepatopancreatic ampulla contains a little channel called the major duodenal papilla. Duodenal, D-U-O-D-E-N-A-L, papilla, P-A-P-I-L-L-A. And the major duodenal papilla then in turn empties those secretions into the duodenum of the small intestine. So, trace out our full pathway for bile. Hepatocytes, bile canaliculi, portal triads, hepatic ducts, converge on right and left hepatic ducts, converge on common hepatic duct, joins with cystic duct to form common bile duct. Common bile duct joins with pancreatic duct at the hepatopancreatic ampulla, which then directs bile into the duodenum at the major duodenal papilla. And that's our full pathway by which not only do we get bile into the duodenum, but also pancreatic secretions. Now, here's an important thing to know. And it goes back to, you might have noticed there was a part in there that I kind of threw in that you didn't hear much about, the cystic duct. Where's that cystic duct coming from? The answer to that is that our liver does not need all the bile it produces at once. Excess bile is stored by the liver in a small sac-like organ called the gallbladder. G-A-L-L-B-L-A-D-D-E-R. the gallbladder. The gallbladder is a little sac found on the inferior surface of the liver. You might recall that the gallbladder is tucked in between the right lobe and the quadrate lobe. The purpose of the gallbladder is to act as a storage sac for bile. Gallbladder is about 8 centimeters long, about 4 centimeters wide. It does expand whenever it fills up. It has a 3-tunic wall rather than a 4-tunic wall. It has a muscularis. Going in order from inside to outside. We have a mucosa lining. The lining of the gallbladder is folded like the lining of the stomach. It has rugae so that it can expand just like the stomach does. Then we have a muscularis deep to the mucosa, and then we have a serosa making up the outside layer of the gallbladder's wall. Now the gallbladder has 2 functions. One, it stores bile. Liver makes more bile than it needs at a given moment. Gallbladder stores it. The gallbladder can store 40 to 70 milliliters of bile at a time. Also, however, the gallbladder concentrates the bile. The gallbladder contains lots of what we call bile salts. Bile salts are mineral salts stored in the gallbladder. They have the effect of concentrating the bile, making it 5 to 10 times more concentrated than it was just in its purely liquid form that it's secreted. That makes it a much more effective emulsifier. So not only is the gallbladder storing bile, it's making it better. Now, when the gallbladder is filled up and the body needs bile, bile will exit the gallbladder through the cystic duct. The cystic duct will then, as you heard, merge with the common hepatic duct to form the common bile duct. So that's an alternate pathway for bile to get towards the small intestines. That highly concentrated bile is extremely useful for making digestion more efficient. So the gallbladder's got a really important job here. Now, some of you might be aware, possibly from painful experience, that things can go wrong with your gallbladder, particularly if you have a high cholesterol diet. One of the dangers is that crystals of cholesterol can precipitate in the bile of the gallbladder. That can lead to the formation of hard, mineralized objects in the gallbladder called gallstones. G-A-L-L, stones, one word. Now, gallstones can just rattle around in there. They can be painful. They can cause irritation to the inside lining of the gallbladder. But the real danger, of course, is that a gallstone will leave the gallbladder and obstruct or partially obstruct the cystic ducts or the common bile ducts. If that happens, you're going to have lots of pain, uncomfortable digestion, inability to fully finish digesting your food because you're not getting enough bile. Fat digestion in particular will become very, very difficult. So gallstones can be quite painful and ultimately can require surgical removal if they are blocking off an important duct. Now, in some instances, if the gallbladder is consistently forming gallstones, or if it has formed a lot of gallstones, basically filled up with them, our only choice might be to remove it. If that is the case, then we have performed a surgery called a cholecystectomy. C-H-O-L-E-C-Y-S-T-E-C-T-O-M-Y, cholecystectomy. That is the surgical removal of the gallbladder, which can obviously solve the problem. Now, the gallbladder is one of those organs you can technically live without. You do not need a gallbladder to function. So you have a cholecystectomy? Yeah, you can absolutely live without that thing. But here's the thing. If you do not have a gallbladder, 2 things have happened. 1, you have lost the ability to concentrate bile. And 2, you've lost the ability to store bile. What that means is your liver can't make as much bile at once. It can still make bile. It's still collecting bile from its hepatocytes in order to get it to the small intestine. But you can't store more, so you can't secrete as much at one time. And you also cannot concentrate the bile, because without the gallbladder to store bile salts, well, that concentration function can't be done. As a result, you have less bile and it's less effective. The upshot of all this is that if you've had your gallbladder removed, you are going to have some digestive problems from that point onward with fats. You're going to have a slightly harder time processing fat-rich meals. Not to say that you can't do it, but it does mean that you can have a little bit of difficulty doing it. A very fat-rich meal can become very uncomfortable for you. But some folks have more problems with that than others. Some don't. Depends on what you eat. Depends on the composition of your individual liver. But you can get away without having a gallbladder. Just might lead to some digestive adjustments. By the way, a thing I didn't mention before, those bile salts. 90% of the bile salts that are produced by the gallbladder are going to travel out of the gallbladder along with the bile that's going out, but most of them are going to be reabsorbed by the duodenum. And the duodenum is then going to send them right back to the liver, and the liver will secrete them right back into the gallbladder. So the same bile salts get used again and again. They leave the gallbladder along with the bile. They're reabsorbed by the small intestine, taken back to the gallbladder. You have to make some. You recollect about 90% of them, so bile salts are continuously produced to replenish them, but you can recapture most of them. All right, so that's the liver. As you can see, it's doing a lot of stuff. The liver is an incredibly busy organ, and it's making a very important digestive compound. It also has a very important role in what happens to the nutrients after they are absorbed by the small intestine. Before they go anywhere else in the body, the liver grabs a hold of what it needs for storage, for nutrient conversion, and so forth. The nutrients the liver doesn't take are then just distributed by the bloodstream to the other tissues of the body that requires them. But there's one other big organ that is helping out here with the finishing of chemical digestion. And that's an organ you've also heard about before, the pancreas. P-A-N-C-R-E-A-S. The pancreas is a long, spongy organ, about 15 centimeters long, so about your hand a little bit more, something like that. The pancreas is a retroperitoneal organ. It is located behind the peritoneum against the posterior body wall. It is found posterior and inferior to the stomach. The pancreas has a head, a body, and a tail. The head of the pancreas is tucked into the U-bend, the curve of the duodenum, like so. The body of the pancreas runs underneath the stomach. The tail of the pancreas, the far end of it, reaches as far as your spleen. Not connected to the spleen, but it gets to that point. Now, the pancreas is an interesting organ because it has both endocrine and exocrine functions. It's part of both the digestive system and the endocrine system. Now, the endocrine functions of the pancreas you already know about. You have heard about them when we talked about the endocrine system back a while back in A&P1. Your endocrine functions of the pancreas are carried out by little clusters of cells called the pancreatic islets, I-S-L-E-T-S. The word islet means little island. Pancreatic islets are scattered all throughout the pancreas. They are also called by an older name, islets of Langerhans, named after the German anatomist Paul Langerhans, capital L-A-N-G-E-R-H-A-N-S. But you might recall our preference in anatomy these days is not to name things after people. We're trying to do away with the old eponymous naming and give things more technical names. So I prefer pancreatic islets. But here, islets of Langerhans, same thing. The islets of Langerhans carry out the endocrine functions of the pancreas, secreting the hormones insulin and glucagon. You might recall those are both metabolic hormones that help us to regulate our blood sugar and our proteins. So pancreatic islets do that endocrine function. It's indirectly related to our body's metabolism. Well, it's directly related to our metabolism, indirectly related to digestion, because basically your body digests the carbs and puts the sugars into circulation. Insulin regulates the levels of sugars. So the insulin will basically tell our cells, hey, this is how you take up sugar, and it will also tell the liver, hey, time for you to lift sugar out of the bloodstream and convert some of it to glycogen. But we are not here to talk about endocrinology. We've talked about endocrinology before. Our interest right now is in the exocrine functions of the pancreas, which involve the secretion of important compounds for digestion. The exocrine parts of the pancreas, which by the way are the majority of its structure, about 98 to 99% of the pancreas is exocrine rather than endocrine. The islets of Langerhans only occupy 1 to 2% of its mass. But the exocrine cells of the pancreas are called the acinar cells. Acinar cells, A-C-I-N-A-R, or acini, A-C-I-N-I for short. And collectively, the acinar cells, or acini, make a group of fluids that we call pancreatic juice. The pancreatic juice is a mixture of several different substances which are essential for the completion of chemical digestion. The bulk of pancreatic juice is the aqueous components, A-Q-U-E-O-U-S, aqueous. The word aqueous means watery or liquidy. The aqueous components is a water-based solution that is rich in bicarbonates. By having high bicarbonate levels, that makes it a basic fluid. Remember that the pancreatic enzymes require a basic environment to operate, which is why we need bile to help neutralize the acid chyme present in the duodenum that was delivered straight from the stomach. Remember, everything entering the duodenum is coming directly out of the stomach. Gastric juice is highly acidic, lots of hydrochloric acid, therefore being dumped right into the duodenum. So bile helps to neutralize it, and the bicarbonate present in the aqueous component of pancreatic juice helps to neutralize it. That makes the pancreatic enzymes have the pH that they need to operate. They operate best in a neutral to slightly basic pH. Now, the other part of pancreatic juice is the enzymatic components. Several enzymes are present in pancreatic juice. Among them, we have pancreatic amylase, P-A-N-C-R-E-A-T-I-C, pancreatic amylase, A-M-Y-L-A-S-E. That is a carb-digesting enzyme. Hopefully you remember amylase refers to a carb-digesting enzyme. We have pancreatic lipases, several of these different enzymes. L-I-P-A-S-E is lipase. Those are lipid-digesting enzymes, as you can probably judge from the name. We have several different proteolytic enzymes, P-R-O-T-E-O-L-Y-T-I-C, proteolytic, which means to break down proteins. Proteo for protein, lytic from lysis, to break down or to rupture. Your proteolytic enzymes are secreted in inactive form, because otherwise the very ducts that secreted them would be digested by them. They only activate whenever they hit an environment of the right pH in the duodenum. And so they remain inactive till they get there. Once they're in that environment, they can begin digesting proteins there, and the mucus-rich lining of the duodenum protects it against being digested by them. And finally, pancreatic juice contains nucleases, N-U-L-E-A-S-E-S, nucleases. These are nucleic acid digesting enzymes. Now, we haven't talked about this much yet, have we? We talk about nutrients. You know, when you look at your nutrient labels on your food, you see proteins, carbs, and fats, or lipids, right? You don't see nucleic acids. But every living cell has nucleic acids. It's got DNA and RNA, folks. You eat nucleic acids just like you eat everything else present in your food. Now, you never see a calorie value listed for the nucleic acids in your food for the very simple reason that there are so few of them that they do not provide a source of energy. In fact, your body does not have a mechanism for digesting nucleic acids to make ATP. They are not a metabolic molecule. But you do need to digest them. So nucleic acids, DNA and RNA, that came in the cells of your food are going to be digested by pancreatic juices. And that's important because that breaks them down into their individual nucleotides, both DNA nucleotides made of deoxyribose and RNA nucleotides made of ribose. Then your body can absorb those nucleotides and use them to build its own DNA and RNA. Those nutrients can be upcycled by your body and used to assemble your own nucleic acids. So we don't think about nucleases in terms of food digestion for metabolism. We're not getting energy from the DNA and RNA of the things we eat, but we are getting raw materials that your cells can utilize. So if you were paying attention there, you probably realized that pancreatic juice contains enzymes that we need to digest proteins, carbs, lipids, and nucleic acids, all 4 of the basic types of organic molecules. Now, this pancreatic juice is going to leave the pancreas by way of the pancreatic duct. Pancreatic duct, as you already heard, fuses with the common bile duct at the hepatopancreatic ampulla, and the collective secretions of both are emptied into the duodenum by the major duodenal papilla. So both bile from the liver and pancreatic juice from the pancreas enter the duodenum together. They help each other to function. The bile helps to neutralize the environment, which makes it easier for proteolytic enzymes to operate. The bile also emulsifies fats, which makes it easier for pancreatic lipases to work on them and digest them. All of these compounds together are essential for completing chemical digestion. Most of the chemical digestion happens right there in the duodenum. You cannot complete it without the secretions of the pancreas and the liver. In particular, the pancreas is vital to this process. Those pancreatic enzymes are the major thing needed to complete chemical digestion. If you do not have those enzymes sufficiently, most of your food will not be digested. It will pass through the small intestine, into the large intestine, and it will exit the body. Which means if you have a malfunctioning pancreas, even if you are eating properly, you can starve to death because your body is unable to completely process those nutrients. You might be aware that if you have pancreatic cancer of the pancreas is of course a very serious issue. Liver cancer, for obvious reasons, is also a very serious issue. And both of those are serious because without both of those things, you can't digest and handle nutrients properly. And people with pancreatic cancer in particular often shed weight extremely rapidly. It's not unusual to lose hundreds of pounds while you are suffering from pancreatic cancer if you have hundreds of pounds to lose, as your body basically cannot get the nutrients that it requires. So, functioning pancreas, super important. You can, as you of course know, I'm sure you all know, that if you're suffering from liver failure or pancreatic failure, we can do transplants. We can transplant a pancreas into you. We can transplant a liver into you. But if either of those organs is not functioning right, you're not going to live long without it. So we don't have an equivalent of a dialysis machine for failed kidneys, for a failed liver, or for a failed pancreas. You go into liver failure, you're going to last about a week, maybe. If you go into pancreas failure, it's all going to depend on how much reserves your body had in terms of the nutrients that it requires. And you can transplant those organs, but the liver is hard to match somebody with for the same reasons as the kidneys, very rich vascular supply. And if you transplant either of those organs, you run into all the problems you always run into with transplantation, namely the risk of rejection. All right, so with the help of those structures, with the help of the pancreas and the liver, the small intestine can complete chemical digestion. That mostly happens in the duodenum. That little 25 centimeters or so of loop at the beginning of the small intestine completes the bulk of your body's chemical digestion. From there, of course, nutrients move on. They get absorbed through the duodenum, the ileum, and the jejunum. I guess duodenum, jejunum, ileum. Jejunum is the primary segment of the small intestine involved in absorption. Ileum picks up things that were not picked up by the jejunum or the duodenum. But presently, we get to the end of the small intestine, where all that is left at this point is a very few nutrients that have not been digested yet, a lot of non-digestible materials such as plant fibers, and some fluids. Electrolytes too, salts and liquids along with them. Now at the end of the small intestine, you hit the ileocecal junction, I-L-E-O-C-E-C-A-L, ileocecal. And the ileocecal junction is going to be the gateway to our next organ in the pathway, our last major digestive organ, which is the large intestine. Okay, so your large intestine, it's large in the sense that it's wide. It is actually shorter than the small intestine, but it is considerably wider than the small intestine, between 4 and 9 centimeters wide, depending on where in its pathway it is. Large intestine is altogether about 1.5 to 1.8 meters in length, so a good bit shorter than the small intestine. Large intestine runs from the ileocecal junction to the anus. It is the last component of the digestive tract. It is responsible for a couple of things. Its functions include, first off, reabsorbing certain materials the body needs to recapture. The large intestine recaptures water from digested material. It recaptures salts and electrolytes that passed out through the small intestines. Secondly, the small intestine has a limited nutrient absorption function. It takes up a couple of nutrients that were not well absorbed by the small intestine, including some that could not be fully processed by the small intestine. Your large intestine actually has a sizable population of microbes, of living bacteria. More than 700 known species of bacteria, in fact, inhabit your large intestine. And a lot of them help us with the digestion of certain nutrients. Certain vitamins, riboflavin, vitamin K, and folic acid are produced largely in the large intestine as a result of bacterial action, and those vitamins are absorbed there. Also, among other things, milk digestion is completed in the large intestine, and the products of milk digestion are absorbed there. You might know that if you're lactose intolerant, that they recommend you eat probiotic yogurts. What happens there is that the bacteria in those yogurts colonize the large intestine and make it a little bit easier to help you digest milk products in the future. That microbiome, as we call it, microbiome, that population of bacteria in the large intestine, is an incredibly important part of your digestive process. You couldn't finish healthy nutrient processing and vitamin absorption without them. And if you don't have enough of them, you'll get quite ill. We don't even know all the effects of the microbiome yet. There's speculation that intestinal bacteria may be related to mood. They might be related to regulation of inflammation. They might even be related to lymphatic function. This is all still being sorted out, but we know that when you don't have enough of them, you don't do very well. These are the same bacteria, by the way, that if they are unleashed into your body cavity, if the large intestine gets punctured, will cause you to get peritonitis and die. They're really comfortable while they're in there. They're bad anyplace else. And by the way, population of about 100 trillion, with a T, 100 trillion bacteria or so in the average large intestine. So we've got bacterial action helping with the processing and absorption of certain nutrients. We have the reabsorption of water and electrolytes. And of course, we have the production and storage of feces. F-E-C-E-S, sometimes spelled F-A-E-C-E-S with the fancy A-E digraph in there. That's an old-fashioned spelling. You don't see that much anymore, but if you ever do see it, that's what it is. Feces, of course, would be the solid waste materials produced by the large intestine and stored until they can be removed. The removal of solid waste from the large intestine is called defecation. D-E-F-E-C-A-T-I-O-N. Now, the large intestine has two main components. First, we have, well, three components, really. First, we have the cecum. Whenever you join the ileocecal junction, the first part of the large intestine you connect with is the cecum. C-E-C-U-M. Older spelling, C-A-E-C-U-M. If you see it spelled that way, it's not wrong. It's just kind of old-fashioned. I prefer the modern spellings of all these things, by the way. The cecum is a sac that extends about 6 centimeters below the ileocecal junction. It's a storage sac that holds things until they move further along, non-digested material. The cecum is larger, by the way, in herbivorous animals. If you're a deer or a cow, you have a much bigger cecum, and certain aspects of fiber digestion happen there. But in our bodies, the cecum does not function in fiber digestion. It just stores non-digested material until it can be moved along. Now, an important clinical consideration with the cecum. Attached to the cecum, dangling inferiorly from it, is a 9-centimeter organ, a little wormy structure called the vermiform appendix. Vermiform, V-E-R-M-I-F-O-R-M. The word vermiform means worm-like. Appendix, A-P-P-E-N-D-I-X. A lot of times we just call this organ the appendix. The appendix is a little structure that dangles from the cecum, and a few years ago I would have told you it was a vestigial structure, that it had no particular function, that it was a leftover from an earlier stage of our evolution. We know better now. The appendix is in point of fact not a digestive organ at all, but it gets discussed with the digestive system because of its location attached to the large intestine. In point of fact, the appendix is a lymphatic organ. It contains permanent patches of lymphatic tissue. As far as we can tell, it functions in the defense of our bodies from pathogens that have managed to get that far. The appendix also contains a colony of bacteria from the large intestine. There is evidence to suggest that if your large intestinal flora, as we often call them, the intestinal flora are those bacteria, F-L-O-R-A, that if those bacteria get depleted for any reason, like you've just been on a serious antibiotic, the large intestine can be recolonized by bacteria coming from the appendix and help it to recover more rapidly. We think that's a thing that's going on. There is disputing evidence on that. But we definitely know that it's got a lymphatic function, that it's lymphatic tissue helping us to fight foodborne pathogens. Now, it's not doing a lot of that. It's a little teensy organ, and like most of our individual lymphatic organs, you don't specifically need it. You can do without a spleen, you can do without tonsils, you can do without any specific lymph node, you can do without an appendix too. And that's good, because you might lose it. If the appendix becomes blocked off, becomes obstructed, or if it becomes infected, then you can begin to suffer from a dangerous inflammatory infection called appendicitis, A-P-P-E-N-D-I-C-I-T-I-S, appendicitis, inflammation of the appendix. Appendicitis can be quite dangerous because if the appendix ruptures, that will release large amounts of bacteria into your abdominal cavity, and you will run a very serious risk of death from peritonitis in the very near future. Now, anatomically, if you have appendicitis, let me get in a position here to show you a little bit. The characteristic symptom of appendicitis is going to be sharp, stabbing, persistent pain in the region of the appendix. Where do you find the appendix? The appendix is located, or you feel that pain, at an anatomical landmark called McBurney's Point, capital MC, capital B-U-R-N-E-Y, McBurney's Point. McBurney's Point is a spot in your abdomen located about 1/3 of the way, working medially, between the superior iliac crest of the hip and your navel or umbilicus. So, if you look right here, right about there is the superior iliac crest. Here is the umbilicus or navel. McBurney's Point is right about there. So that is where you will tend to feel the pain associated with appendicitis. Sharp, stabbing, persistent pain, often accompanied by nausea and either diarrhea or constipation as possible, honestly. If you are experiencing that kind of pain, don't mess around. Don't wait to see if it gets better because, again, a ruptured appendix is nothing to sneeze at and can be life-threatening. Now, the unfortunate thing here, if you are a woman, is that the pain from appendicitis is located in about the same spot where you're going to feel pain from ovarian cysts. So if you're suffering from ovarian cysts, you're going to be feeling sharp, steady, persistent pain and nausea from that right around that same area. Granted, if you're having ovarian cysts, you should probably get that dealt with too. So if you've got sharp, persistent pain from right around that vicinity, go get it checked. You really, really don't want to mess around with it. All right, so that's the appendix. Whenever you're suffering from it, easiest way to deal with it is an appendectomy. A-P-P-E-N-D-E-C-T-O-M-Y. Appendectomy, surgical removal of the appendix. Like I said, you don't need it. You can cut it out without any harm. All right, but cecum, inferior-most portion of the large intestine. It's the entry point from the ileocecal junction. From the cecum, we are going to move on to the largest portion of the large intestine, which is the colon, C-O-L-O-N. The colon is between 1.5 and 1.8 meters long. It's most of the length of the small intestine. Got 3 parts. Coming up from the cecum is located in the lower right corner of your abdomen. we are going to have the ascending colon, A-S-C-E-N-D-I-N-G. Ascending colon travels up the right side of the abdomen, running from the cecum to the liver. Then we're going to have a bend called the right colic flexure. Colic, C-O-L-I-C, flexure, F-L-E-X-U-R-E, the right colic flexure. That leads us, so we got ascending colon right up here, like so. Then you're going to have the transverse colon. Transverse colon is going to go horizontally across the abdomen, running from right to left, runs from the liver to approximately the location of the spleen. At the spleen, you're going to have another sharp bend, the left colic flexure, also called the splenic flexure, S-P-L-E-N-I-C for the spleen. And then you're going to have the descending colon, which runs down the abdomen from the left colic flexure or the vicinity of the spleen to approximately the top of the pelvic girdle, right about here. From there, you are then going to have the sigmoid colon, S-I-G-M-O-I-D. The word sigmoid means S-shaped. It's going to curve down and up and down again. Sigmoid colon goes across the abdomen that way. Sigmoid colon is going to extend medially and inferiorly and terminate at the rectum, which is going to be the final segment of the large intestine. We'll get to that in a few minutes. So we got ascending, transverse, descending, sigmoid. The ascending and descending portions of the colon are both retroperitoneal. The transverse and sigmoid portions of the colon are covered by the peritoneum. Now, if you look along the length of the large intestine, it's going to have a very kind of little bulgy, puckered appearance to it. You know, it's going to have like little poof, poof, poof kind of lumps. There's a great technical terminology for you there. Let me just draw you a quick diagram. All right, my pen is dying. Figures. All right, but you're going to find along the length of the large intestine, you're going to have these little puckered pouches all along its length called haustra, H-A-U-S-T-R-A. You're also going to find a long, narrow band of longitudinal smooth muscle along the length of the colon called the teniae coli, T-E-N-I-A-E, coli, C-O-L-I. So that is going to be this band of smooth muscle that runs along the full length of the colon. Those little puckered pouches, the haustra, all along its length. If you look at the inside lining of the colon, it's going to have a four-tunic wall like most of the digestive tract. Its mucosa is not folded the way that the mucosa of the stomach and the small intestine are folded. You will find that in the lining of the large intestine, there are numerous straight tubular glands, little tiny straight tubular glands called the colic crypts, C-R-Y-P-T-S. Those are mucus-secreting glands for the most part. They also have some absorption function. The things that are absorbed by the large intestine are largely going to be absorbed by the colic crypts. Now, we're going to go up ascending, across transverse, down descending. Now, moving things along the length here, as we are transporting things along the length of the small intestine, its major secretion is going to be mucus, lots and lots of mucus. Couple of functions. One, lubricates the walls to prevent abrasion from the material moving through. Two, helps to stick feces together, to bind together the waste materials of feces. Along the way, we're also going to encounter lots of bacteria, which, as you know, help us with dairy digestion, processing certain vitamins along the way. Now, those bacteria also, I should point out, as they're doing their thing, as we're moving material along the large intestines, they are going to be doing a couple of different functions. The chief one is going to be making feces. Feces are formed in the colon. This is the solid waste material of the digestive system. The largest bulk of feces is going to consist of undigested material from your food, the most significant component of which is plant fibers. Your body is not really good at digesting plant fibers. Also present in feces, you will find bacteria that get caught up in them. You will find dead epithelial cells shed from other parts of the body. You'll find a small percentage of water, about 4% of the water that leaves your body leaves via the feces, but in general, most of the water coming into the large intestine is going to be reabsorbed. Now, once material gets into the large intestine, it's going to be there for a while. On average, it takes 18 to 24 hours from the time stuff lands in the large intestine till the time that it makes its way out. Now, large intestinal movements are going to be a little different than those of what we saw in the small intestine. You're going to have bands of muscle along the length of the large intestine found at the places where those haustra pucker inward. We call those circular muscles. And you're also going to have the teniae coli along its length. During what we call large intestinal movements, also known as mass movements or bowel movements, what's going to happen is the circular muscles are going to contract, pinching the haustra inward, and the teniae coli is going to contract, pulling the intestine upward. This is going to have the effect of constricting and shortening the colon, slowly mixing up its contents and pushing them upward and along. The puckered walls of the haustra will temporarily store material to prevent it from falling back down until it's pushed a little bit further up in the line. As the material is moving through, it is being slowly mixed with mucus. It is being compacted into feces. Water and electrolytes are being reabsorbed along the way, largely by the colic crypts. The bacteria in the colon are busily working on nutrients. They are digesting some materials that were not previously digested. This is going to help us break down milk sugars, among other things. It's going to help us to break down certain vitamin products that were not fully broken down in the small intestine, which can then be absorbed. Vitamin K, folic acid, riboflavin, as I mentioned before. We've also recently learned that a small amount of carbohydrate and fat digestion also happens in the colon. Those bacteria do break down some plant fibers and fats that were not fully processed by the small intestines. In particular, they break down some of the plant fibers that your body is otherwise incapable of digesting, but not all of them, just a little bit of them. I should point out that a byproduct of that bacterial metabolism is not just going to be vitamins and nutrients that your colon can absorb, but it's also going to be a small amount of bacterial gas, primarily methane gas. The gases secreted by the bacteria in the large intestine are collectively called flatus, F-L-A-T-U-S. Flatus is periodically released from the body through the anus to the amusement of 12-year-olds everywhere through a process called flatulence, F-L-A-T-U-L-E-N-C-E. And that's just a byproduct of bacterial metabolism. So ultimately, though, we're retaking up the stuff that we didn't want to leave. We're reabsorbing water and electrolytes, putting them back into circulation in the body. The bacteria are releasing some useful things that we can also absorb, vitamins and a couple of digestion products. And everything else is being compacted into feces, dead cells, bacteria, non-digestible fibers, wee little bit of water, and mucus to bind the whole thing together. Now, our movements through the large intestine, the mass movements as we call them, are going to be peristaltic movements. It's a slower peristalsis than what you saw in the stomach or the small intestine. They happen several times a day, stimulated by two primary reflexes. There is the gastrocolic reflex, G-A-S-T-R-O-C-O-L-I-C. The gastrocolic reflex begins mass movements whenever food arrives in the stomach. This is basically the body's way of saying, hey, there's another meal coming along down the pipeline up here. It's going to be here in a few hours. You better start clearing things out. So when food arrives in the stomach, the gastrocolic reflex stimulates mass movements. This is why sometimes you find yourself having to defecate after a big meal. It's not because that meal went right through you. It's because that was a meal from yesterday that is being pushed along. There is also the duodenic reflex, D-U-O-D-E-N-I-C. The duodenic reflex, as you might guess, is whenever material chyme from the stomach arrives in the duodenum, because that's further saying, hey, another meal moving along here, it's done with the stomach part, it's in the small intestines, it's getting to the large intestine in 3 to 5 hours, therefore helping to clear things out by moving stuff along. So the arrival of those things is most likely to stimulate mass movements. Mass movements can also happen in response to the smell or taste of food. Helps to stimulate, gets the digestive process going, as you already know, the psychological effect of that. Says, hey, there might be a meal coming, I better get ready. But stomach distension, stretching of the stomach as you're eating, will stimulate this. The more the stomach gets distended, the greater the likelihood of a mass movements. Most common time is right after breakfast. Your first meal of the day is typically when your first mass movement of the day will happen. Once mass movements begin and stuff begins moving along the colon, they're going to last for 10 to 30 minutes. You're going to have 10 to 30 minutes of slow peristaltic contractions, gradually shifting stuff along the colon. After that, it's going to stop for 8 to 12 hours. And then more mass movements will happen again later, usually depending on when you're eating and so forth. As we're moving along, we are compacting the material into feces, soaking up water and salts and so forth. Important thing to note here. If the lining of the colon becomes irritated, you've eaten something that is noxious to you, you've got an infection of some kind, or sometimes as a consequence of stress for reasons that we're not clear on. But if the lining of the colon becomes irritated, it might become a little inflamed, a little bit of colitis, but an irritated colon does not effectively reabsorb water and electrolytes. A consequence of that can be that you end up passing a lot of water along with the feces, which you all know is diarrhea. D-I-A-R-R-H-E-A, diarrhea. So that's the excretion of excess water with the feces because the colon lining was irritated and it was not properly reabsorbing the water along the way. Diarrhea is an occasional consequence. Everybody suffers from colic irritation, but if it is chronic, it can be life-threatening. Several diarrheal illnesses like cholera and C. diff can kill you because you can die of dehydration and electrolyte depletion because normally your colon is reabsorbing those things, but an irritated colon cannot effectively do so. Cholera, by the way, C-H-O-L-E-R-A. It's not a disease we really hear about in the United States much anymore. It's largely been cleared up by improved public sanitation and sewage systems. But in developing countries that don't have those systems as well as we do here, cholera is very much a dangerous condition that still claims a lot of lives. And C. diff, of course, you probably know, is one of our lovely antibiotic-resistant bacteria that can cause significant diarrhea and can cause dehydration and electrolyte depletion, which can kill you. So diarrhea in the short term happens to everybody from time to time. If you end up with a so-called stomach flu is not actually, in fact, a flu. It is usually a viral or bacterial infection in the digestive tract, sometimes a norovirus or other, minor digestive bugs. But either way, that can often cause irritation that gives you a couple of unpleasant days of diarrhea before your body basically excretes and defeats the infection. So, thing that happens from time to time. Now, assuming everything's going the way it should, we get to the end of the colon after 18 to 24 hours. At the end of the colon, the sigmoid colon is going to empty into the rectum, R-E-C-T-U-M. The rectum is a straight tube with a very thick muscularis. At the terminus of the rectum is the anal canal, A-N-A-L. Anything that is anal pertains to the anus. Anal canal is 2 to 3 centimeters long. and it is the bridge between the rectum and the anus, the outside opening of the body. Now there is a sphincter, a muscular sphincter at either end of the anal canal. There is an internal anal sphincter, which is a smooth muscle sphincter that you have no voluntary control over. There is an external anal sphincter located at the anus itself, ANUS. That is a skeletal muscle sphincter over which you do have voluntary control. Pressure of feces against the rectum can eventually cause the internal anal sphincter to open, pushing material down into the anal canal. That then puts pressure on the external anal sphincter. But the external anal sphincter is a voluntary one. And that brings us to the defecation reflex, DEFECATION, the elimination of solid wastes from the body. Now, the defecation reflex is stimulated largely by distension of the walls of the rectum. That is, feces accumulate in the rectum. Its walls get more and more stretched, and that begins sending a signal. The stretch receptors in the wall of the rectum stimulates a local reflex, causes the internal anal sphincter to relax. It's a smooth muscle sphincter. You have no control over that one. Feces then begin to move down into the anal canal. That, in turn, is going to cause stretch in the anal canal, which will send a signal to the defecation center. The defecation center is a nerve center located not in the brain, but in the spinal cord. Specifically at the base of the spinal cord, you will find the conus medullaris, C-O-N-U-S, conus medullaris, M-E-D-U-L-L-A-R-I-S, conus medullaris. This is the inferior end of the spinal cord. So the spinal cord is largely the regulator of defecation. Even if you are clinically brain dead, the spinal cord will control when defecation happens. So anal canal gets stretched, sends a signal to the conus medullaris. Conus medullaris then sends a signal back to the colon, which causes further relaxing of the internal anal sphincter and contraction of the sigmoid colon and the rectum. That tends to push feces further down and puts pressure against the external anal sphincter. This, in turn, sends a signal to the cerebrum saying, hey, 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 you need to defecate. That's the signal when you feel the need to release solid wastes. Now, that creates an awareness. That awareness can be ignored. It's easier to ignore the defecation reflex than it is to ignore the micturition reflex. It is easier to delay defecation than it is to delay urination, for the simple reason that if you ignore it long enough, the muscles will begin to relax, feces can continue to pile up in the sigmoid colon. And after a few minutes, normally that reflex will relax. But it's going to come back. Pressure is going to continue, and eventually, as with urination, eventually the pressure will give, and eventually you will have to release the feces, whether you want to or not. In general, it is better not to delay defecation if you can avoid it, simply because, like every part of your digestive tract, it begins building up habits after a while. If you get in the habit of delayed defecation, your body does not do mass movements as much, which can lead to persistent constipation and infrequent emptying of the colon and backup of feces, which can be quite problematic. Now, the thing about this is that the external anal sphincter is a skeletal muscle sphincter. It's under conscious control. You have to choose to open it. Little children, little kids and babies, don't know how to do this yet. And as a result, whenever they feel that pressure, they're just going to let it go. Typically, humans learn control of the external anal sphincter before the age of 2, or between the ages of 2 and 3 at the latest. That's about the norm here. And that means that after a while, you can control when you need to defecate and delay defecation until you get to a place where you can do so. Sometimes, if feces are heavily built up, conscious muscular contraction of abdominal muscles will be needed to help squeeze them completely out of the large intestine. Yet another reason not to avoid defecation. It becomes more and more difficult to remove feces if you have been delaying for a while because there's a larger buildup and more muscles will be required to make it go. All right. Now, how often is enough? This is a thing you'll hear questions about periodically. How often should a healthy adult be defecating? Most healthy adults with a normal diet will need to defecate at most every day or every other day. Depending on what you're eating, you may need to defecate more. A large amount of plant fibers in your diet may lead to more frequent defecation because you've got a larger bulk of feces from non-digestible cellulose. Now, some people will defecate more or less. Generally speaking, it's not a problem unless you're defecating very frequently and passing out lots of liquid along with the solid material, in which case that's obviously a problem. The other end problem is if you have constipation, C-O-N-S-T-I-P-A-T-I-O-N. Constipation is clinically defined as 2 bowel movements per week or fewer. So if you're defecating twice a week or less, that is clinical constipation. Anything more frequent than that, you're probably okay. Possible causes of constipation are quite frequent. Delayed defecation. You delay defecation long enough, constipation becomes likely. Dehydration. Less fluid volume makes it harder to move feces along. You need some fluid in there. Irritation of the colon by a variety of diseases can cause constipation. Clinical depression can cause constipation. We don't know why, but there's a strong link between the two. Pregnancy frequently causes constipation, side effect of hormonal changes. Changed dietary habits. If you've begun eating at different times or eating a different mixture of foods, then a lot of times constipation will result. Sometimes actually diarrhea can result too. If the colon is obstructed, most often this means a blockage from a tumor or something like that can result in constipation. Or if there are serious injuries to the lower colon or the spinal cord, you may find that there is damage to the defecation reflex that makes it harder to send that signal of the need to defecate. Constipation is often treated by dealing with the underlying cause or by administering a laxative drug, L-A-X-A-T-I-V-E. Laxative drugs stimulate mass movements of the colon in order to propel things along. All right, now to close out this chapter, let's talk about a couple of intestinal pathologies, because there are several pathologies of the digestive tract that deal primarily with the colon and or with the small intestine. And I'm about to tell you right up front, some of these pathologies we do not understand well. Some of them have contradictory symptoms where they cause both constipation and diarrhea, for example. And a lot of them we don't know the causes of. All of them seem to have a strong psychological link. They are more common in people who are stressed, anxious, or depressed. We do not know why. Well, all except for this first one. The first one and the most significant colic pathology that we need to discuss is colon cancer. Colon cancer is the second most lethal cancer in the United States, claims approximately 55 to 60,000 lives every year. Now, colon cancer is strongly tied to diet. In particular, colon cancer is a frequent result of diets that are low in fiber and high in fats and processed meats or red meats. These materials produce feces of less bulk. If you're having a low fiber diet, and most Americans do, then you will not have sufficient bulk in your feces. Likewise, fat-rich diets and processed meat-rich diets do not produce as much bulk of feces as well. A consequence of this is that your colon has to strain harder to eliminate feces when they take up a smaller volume. It's not as easy to squeeze them along. Straining of the colon can lead to irritation of its lining, which causes the development of colon polyps, P-O-L-Y-P. Polyps are raised, irritated patches in the colon. Some of them are nipple-like polyps that protrude from the wall of the colon. Some of them are so-called carpet polyps, which are lower, sort of dome-shaped areas on the colic wall. Colic polyps can be quite painful. They can be irritating. They can lead to more frequent diarrhea or constipation. But more importantly, colic polyps are often the precursors to colon tumors. So there is a direct link between a low-fiber diet and colic tumors. Most Americans are not getting as much fiber as they require. 25 to 30 grams of fiber a day is generally recommended. Men in particular are often prone to low fiber diets. And a lot of this is because we don't eat a lot of fruits and vegetables, which contain a lot of plant fibers. And most of our bread products that we are eating are not whole grain bread products. They're white bread products that have very little fiber in them. Whole grain products have all the good fiber in them. So make sure you're getting enough fiber. If worst comes to worst, take a fiber supplement. You're doing your colon a favor. Colon cancer is so lethal, in large part because the digestive problems it causes, like constipation and diarrhea, have so many other potential causes that a lot of times they go unnoticed, that you don't realize anything is wrong until the cancer has become quite advanced. And therefore, a lot of times colic cancer has either advanced to the point where it can't be stopped or it has metastasized before you know you have it. Best tool, as with many cancers, is prevention. Eat a high fiber diet, make sure that you're not consuming too much saturated fat or processed meats. That should help out with most cases. About 75% of colon cancer cases are linked to low fiber diets. Men are more at risk than this from women for reasons we are not clear on. Men get colon cancer about twice the rate of women, but doesn't mean women are immune. Big preventative, once you pass the age of 40 or so, that's when the risk of this really begins to go up. Once you get to about your mid-40s, you're going to want to get an uncomfortable procedure from time to time called a colonoscopy. colonoscopy. A colonoscopy. It's colonoscopy. I don't remember if I put the O in there. Colonoscopy. And this is where we run a probe through the anus and up into the colon to inspect the walls of the colon looking for polyps. In a lot of colonoscopies, polyps can be surgically removed by the same probe that's going up in there at the same time that it's happening. Nobody likes a colonoscopy, not a fun procedure, but once you're in your early to mid-40s, especially if you have a familial history of digestive disorders, you're going to want to get a colonoscopy every couple of years or so just to make sure that you're watching out for things before it becomes a serious problem. Now, some other somewhat more mysterious pathologies of the colon. There's a condition called Crohn's disease, capital C-R-O-H-N. Crohn's disease is a localized inflammatory disease that causes degeneration of the digestive tract. It mostly affects the proximal end of the colon, but it can also affect the ileum of the small intestine. Intense inflammation in the affected regions leads to diarrhea, sharp abdominal pain, persistent fever, and severe weight loss as a consequence of insufficient nutrient absorption. The causes of Crohn's disease are unknown. It is a progressive condition that will gradually destroy more and more of the colon. It tends to come episodically. You'll be okay for a while, and then it'll flare up again for a little while. Then you'll be okay for a while and flare up again for a while. During those flare-ups, last anywhere from days to weeks, you're likely to lose a lot of weight. A friend of mine who has Crohn's disease during one of his more serious flare-ups shed a dramatic 90 pounds within a couple of weeks. Now, we don't know the cause of Crohn's disease. It appears to be an autoimmune thing, inflammatory attack on the tissue of the colon itself. We are not sure why it happens. There are anti-inflammatory therapies that have been attempted to slow down its progression, but there is presently no cure. If enough of the colon is destroyed, you may not be able to pass feces properly, and you may then have to have a procedure called a colostomy, C-O-L-O-S-T-O-M-Y, which is the insertion of an external bag for the collection of feces, since you can't move them along the colon yourself. Do not know why it happens. Similar condition, actually often confused with Crohn's disease in the early stages, is ulcerative colitis. Ulcerative, U-L-C-E-R-A-T-I-V-E. Colitis, C-O-L-I-T-I-S. Not all colitis is ulcerative colitis. Colitis is any inflammation of the colon. A lot of times that's a temporary condition born from a bacterial or viral infection. Ulcerative colitis is characterized by inflammation, edema, and hemorrhaging within the large intestine and rectum. A lot of the presentation of ulcerative colitis is similar to that of Crohn's disease. Intense pain, fever, severe weight loss. Ulcerative colitis is also often characterized by frequent diarrhea and by the presence of blood in the feces because of bleeding into the colon. Could also lead to anemia with frequent enough colic bleeding. As with Crohn's disease, this tends to be a progressive condition that gets worse and worse over time and can eventually lead to the inflammatory destruction of portions of the colon. Cause is unknown. We don't know if it's caused by the same thing as Crohn's disease or if they're just presenting similarly. Then we have the really strange one, IBS, irritable bowel syndrome. Irritable, I-R-R-I-T-A-B-L-E, bowel, B-O-W-E-L. The word bowel is kind of used synonymously with colon in older texts. And disease, irritable bowel disease or irritable bowel syndrome, often called IBS for short. Older name for this that you don't hear as often anymore is spastic colon, S-P-A-S-T-I-C. Spastic colon, yeah, so it's the same thing as irritable bowel disease or IBS. IBS is characterized by abnormal intestinal movements, either diarrhea or constipation, often alternating between the two. Characterized also by pain in the lower left quadrant of the abdomen, opposite side from where you would get appendicitis. So you've got episodes of lower abdominal pain, either diarrhea or constipation, sometimes gastroparesis. We have no idea what causes IBS. And the weirdest thing about IBS is that it is not associated with any specific anatomical abnormality. There is nothing we can look at in the colon and say, oh, right there, we see irritation in that spot, or we see inflammation in that spot. There is no actual visible abnormality responsible for IBS. There's nothing apparently visibly wrong with the colon. It just isn't working right sometimes. Seems to be a chronic condition. There seems to be a familial tendency to develop it, tends to run in families. It is strongly linked to psychological stress. You're more likely to have episodes of IBS whenever you're anxious or stressed. And so people with anxiety and depression often experience IBS as a comorbidity. For reasons we are also not clear on, it afflicts women approximately twice as often as men. So men get colon cancer more, women get IBS more. IBS is unlikely to kill you. It's uncomfortable, it's unpleasant while it's happening, tends to come episodically and go away and then come back again. We have no idea why it happens. We have no idea what to do about it when it does happen, aside from trying to make you comfortable at the time. If you're living here in Pittsburgh, Magee Health System in Pittsburgh, Magee Women's Hospital, actually has a center dedicated to IBS research where they're trying to investigate causes of the disease. All right, folks, now this has run a bit longer than I normally do my lessons, but I honestly only had about 20-25 minutes left of material after the normal point when I would have cut short, so I decided it was probably a good idea to just carry through and finish this out rather than posting a really teensy lecture afterwards. So that concludes our lessons on the digestive system. I hope you have enjoyed. Hope you all have an excellent night. Talk to you soon.